Okay, guys, I wanted to spend some time going over our results from the PCR lab. And so I have the results here from table 4, because I think they ended up, for whatever reason, with the best looking gel. At least it's the easiest to see the bands. So if we go back to our directions and we take a look at what we should see inside of these lanes. Remember that when we loaded the gel, okay, I gotta go up here and find the, okay. So when we loaded the gel, our standard was in the first lane, the next 3 were controls, and then we had your actual student samples. So if we go back over here, the one that is the molecular marker, the DNA, is the one that has the most marks. And so in this case, it's this lane right here. So this is lane number 1. And so this lane to the farthest left corresponds to the DNA standard over here. So that is lane number 1. And so the purpose of this is that standard has fragments of DNA that are a known size. And so if we wanted to figure out how large each of these fragments is, we can compare it to the known values over here and then calculate how big these fragments are. For the test that we did, we were just looking to see what kind of fragments exist in the sample, not their size necessarily. So the next 3 lanes are the control. So one of them is the positive positive, one is the positive negative, and one is the negative negative. Remember that for those controls, if it says positive positive, it's not like that means that the positive positive has something and then the negative negative tube is empty. It just means these are like the different alleles. And so this would be like homozygous recessive, this is homozygous dominant, and then this is like heterozygous. And so if we go back over here, one of these is going to be homozygous positive, one of them is homozygous recessive, or positive positive, negative negative. You can go back and figure out which one they are, but that's what these are. So these are your 2 homozygous samples. The heterozygous individual, the one that has 1 positive and 1 negative, would be here. And again, these are controls. So this tells us that if you are heterozygous, you should have bands that look like that. And if you are positive positive or negative negative, you should have bands that look like these 2, because that's what they are. They're the homozygous controls. And so these first 4 lanes are just to tell us that the experiment worked at all. So if we had the alleles present in our sample, they should have run out on the gel, and they should correspond to the fragment sizes that we have over here in the standard. The next 3 lanes are the students. And so in this particular group, they had 1, 2, 3 students at least. There could have also been a 4th student that, maybe it didn't come out or something. But I can see data for at least these 3. And so if we look at this individual over here, you see that there's 2 bands. You'll also notice it looks like there's other bands. I mean, I kind of see a mark over here, and maybe I see a mark over here. And it could be that basically that's contamination. You'll notice even over here for the standard, we had other faint markers that were showing up. And so this can be pieces of DNA that didn't completely synthesize, or contamination in some other way. But this individual would be like heterozygous. This one looks like they're homozygous with the control that matches over here. And then this individual looks like they're also homozygous with the control that matches over here. And I'll let you guys figure out which one's the larger and which is the smaller fragment size, which really isn't that complicated. It tells you down here which one of those, down in here, which one you would expect to be smaller or larger based on the positive or negative. And so as long as you see fragments in your student samples, and those fragments match your controls, your experiment worked. For some of you guys, if I come back over here, I'm just gonna pick another table. I'm not trying to pick on anybody in particular. I'm just gonna, go look at some other table. Let's just look at table 1. It's the first one on the list. And we look at their data. When I say that, someone's experiment worked more than another, all that we're really talking about is how well we can see the bands. And so if we look at these other data, I don't know why this one's taking so long. Jeez. Okay, here, again, the data is similar. We got the same sort of thing. It's just that we have much more faint sorts of bands. That could be because the PCR reaction didn't perform as well, and so there's less DNA to stain. It could be just the staining itself, for whatever reason, did not work as well, and so even though there's DNA in there, we just don't see it. But this is just an example of how we got other results, and we can also figure out which one is the marker, the controls, and the student lanes in these, just like the other ones, and figure out who's homozygous dominant, who's homozygous recessive, and who's heterozygous. And so overall, everything worked. That's good news.